Hello, and welcome to Science with Mr. Morgan. In today's episode, we're going to be learning about DNA, RNA, transcription, and translation. So to start, what is DNA? Well, DNA is what makes up our chromosomes and stores our genetic information. Your DNA is different than my DNA, which is different from their DNA, and so on and so forth. It's kind of like a blueprint for living organisms. It's a set of instructions that tells your cells what to do. So, what does DNA stand for? It's an acronym that stands for deoxyribonucleic acid. The deoxyribo portion stands for deoxyribose, which is the sugar found in DNA. The nucleic acid portion represents macromolecules that store information. So, these nucleotides are the basic building blocks of nucleic acids, and DNA is made of very long chains of these. The smallest DNA molecules have over 20 million nucleotides. Now imagine how many more are in larger DNA molecules. Each of these nucleotides is composed of three main parts, which are a phosphate group, a 5-carbon sugar, in DNA is deoxyribose, and a nitrogen-containing base. Now there are 4 of these nitrogen-containing bases in DNA. They're adenine, which is represented with a capital A, guanine, which is represented with a capital G, thymine, which is represented with a capital T, and cytosine, which is represented with a capital C. Each of these bases have specific pairs. Adenine, or A, only pairs with thymine, or T, and vice versa. Cytosine, or C, only pairs with guanine, G, and vice versa. Adenine and thymine never pairs with cytosine or guanine, and cytosine and guanine never pairs with adenine or thymine. All of these nucleotides come together to make up a specific code known as the genetic code. These various sequences of your nucleotide bases make up the genetic code of your cells, and there are many different combinations possible. Thanks to the work of scientists Rosalind Franklin and Maurice Wilkins, two other scientists by the name of James Watson and Francis Crick were able to piece together the structure of DNA in the 1960s. They determined that DNA is made of 2 strands of nucleotides formed into a double helix. If you've ever seen a spiral staircase, it kinda looks like that, or at least that's what I'm referring to-ish when I say double helix. There are sugar and phosphate groups on the outside part of the helix, and paired bases connecting the 2 strands on the inside of the helix. Now I know I mentioned this briefly, but let's delve more into base pairing. Remember that base pairing refers to adenine, thymine, guanine, and cytosine, at least in the case of DNA, which will be important to remember later. These bases do not randomly pair. A biochemist by the name of Erwin Chargaff found that the percentage of adenine in a strand of DNA always equaled the percentage of thymine in the same strand. After a bit more research, the same was found for the percentage of cytosine with the percentage of guanine within the same strand. So adenine and thymine always equaled the same amount, and cytosine and guanine always equaled the same amount. It was determined that these were complementary bases, which are bases that always pair together. They complement one another. Adenine binds with thymine, and cytosine binds with guanine. This information can be used to determine what the other DNA strand reads if you only know one side. If we know half of the DNA strand reads AATCGGCAT, then we know that the other strand has to read TTAGCCGTA. But what holds these bases together in a pair? It's hydrogen bonds. Now, let's take a step away from the bases and instead talk about DNA replication instead, which we'll get into in a different episode in much greater detail. DNA needs to copy itself every time a new cell is created, and the new cell needs to have DNA that is exactly like the rest of your cells to make sure that the cell doesn't malfunction. This is where DNA replication comes into play. It occurs during the S phase, aka synthesis phase, and synthesis means making, of the cell cycle before mitosis or cell division. Wondering what those things are? Don't worry, we cover them in another episode like I just mentioned. To replicate, a special enzyme called DNA helicase breaks down the hydrogen bonds holding the bases together, which causes the DNA helix to unwind, kind of like a zipper. This leaves 2 single strands of DNA, which act as a template for the new strands. After that, another enzyme called DNA polymerase reads the template and uses it to build the new strand of DNA using new nucleotides and joining them together into the DNA double helix shape. We started with 1, but now we have 2 DNA molecules, and each molecule is made up of 1 old strand from the original DNA and 1 new strand. All that being said, DNA isn't the only nucleic acid found in cells. It has the instructions to create proteins, but DNA itself can't make the proteins on its own, so it uses special messages to do so. And these messages are single strands called RNA, which stands for ribonucleic acid, which describes how to make the proteins our cells need. There are 3 types of RNA in protein synthesis, and they are messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Now, all 3 of these are made up of nucleotides, just like DNA has nucleotides. Remember, the nucleotides in DNA are adenine, guanine, cytosine, and thymine. In RNA, the nucleotides are mostly the same, but there's one big difference. RNA does not use thymine. Instead, uracil is used, which is represented with a capital U. So to recap, the 4 nucleotides for RNA are adenine, guanine, cytosine, and uracil. So now that we've established what RNA is, let's take a look at the specific kinds, starting with messenger RNA. Messenger RNA, or mRNA, carries the instructions for protein synthesis from the nucleus, where the DNA is located and the mRNA is produced, to the cytoplasm outside of the nucleus. The mRNA is the instructions for proteins. Ribosomal RNA and transfer RNA are both involved in putting the amino acids in order to make the protein itself. rRNA becomes part of the ribosome, which is where proteins are synthesized. tRNA transports an amino acid to the ribosome so it can be added to a growing chain of amino acids during protein synthesis. There are a lot of tRNAs because each tRNA is specific to an amino acid, and these amino acids are specific to the corresponding tRNA during this process. Let's delve a little deeper into protein synthesis, but first, let's pause and recap what we've talked about with DNA versus RNA. So DNA, remember, is a double-stranded molecule in the shape of a double helix that contains a deoxyribose sugar and uses the 4 nucleotides adenine, thymine, guanine, and cytosine. On the other hand, RNA is a single-stranded molecule that contains a ribose sugar and uses the 4 nucleotides adenine, uracil, guanine, and cytosine. All right, back to our regularly scheduled program with protein synthesis. Proteins are made up of amino acids, like we mentioned. There are a limited number of amino acids in total, specifically 20, but there are many ways that these can be combined. Monomers, which are building blocks, and in this case, amino acids for proteins, bind together to form polymers, which are long chains, and in this case, the proteins themselves. Now, it's important to note that monomers are not always amino acids, and polymers are not always proteins. But like I mentioned, in regards to protein synthesis, this is what we are referring to. Each strand of DNA has many separate sequences known as codons that code for a specific protein. Within each human cell, there are approximately 22,000 genes, which means that they could code for a lot of proteins. Well, they could if they were all turned on, but most of them are turned off and not actively being used by all cells in your body at all times. There are many different cell types throughout the body, and each of these different cell types have different genes turned on, which is known as gene expression. This means that the different cell types make different proteins, depending on what genes are turned on. And it's because of this that the terms protein synthesis and gene expression are often used to reference the same thing. Now, the body itself uses a lot of proteins for a lot of different things, including creating muscles, controlling chemical reactions, transporting essential molecules like oxygen, and performing lots of other important tasks within the body. Protein synthesis is the creation of proteins, but to further explore how it's done, we have to look at what's known as transcription and translation. Transcription is the process of constructing an mRNA molecule from DNA. Remember that DNA is the instructions, but it can't leave the nucleus, so it has to send the instructions out into the cell via mRNA. Transcription is similar to DNA replication, but single-stranded RNA is produced instead of double-stranded DNA. This means that only one strand, or 1/2 of the double helix of DNA is used as a template. The DNA double helix is unwound, and then the enzyme RNA polymerase builds the mRNA molecule using the unwound DNA as a template. Like we've already talked about, the production of mRNA follows the same base pairing rules as the replication of DNA. However, adenine binds with uracil instead of thymine. So in order to be transcribed into mRNA, genes must be turned on or expressed. Your body will not transcribe genes into mRNA if they are off. After this transcription occurs, the newly formed mRNA undergoes a process known as translation. Translation is the process of reading the code of mRNA to make a protein. Specifically, mRNA is translated from the language of nucleic acids, aka nucleotides, to the language of proteins, aka amino acids. Now earlier I referenced something called codons, which are sets of 3 bases. These are read in the ribosome, and each codon codes for a specific amino acid. Once the mRNA has traveled from the nucleus to the ribosome, the tRNA then reads the codon and brings a specific amino acid to attach to the growing chain of amino acids. The anticodon, which is the part that is complementary to the codon, binds to the codon on the mRNA. And once this happens, the tRNA is released from the amino acid. This whole process repeats until it gets to UGA, UAA, or UAG, which are known as stop codons. These code for protein synthesis to stop and tell the ribosome to let go of the newly created protein. And that brings us to the end of today's lesson. Until next time.